All right, so hello everyone. My name is Ty Wilson. I'm an undergraduate researcher in my senior year here at USU. My mentor is Dr. Mita Kondal. We mainly do plant microbe interaction studies, but today I want to talk about two major projects that we've been working on. And kind of the premise of them is that with the environment, the earth kind of shifting, and yet we've got saline levels in the soils going up as well. We're having less water being available. I mean, we've been in a drought for quite a number of years now. There's all sorts of different things that are changing. And on top of that, with population levels increasing, we're needing as well to meet those demands, those food demands. And so part of what we do is try to analyze and try to figure ways that we can provide buffers, provide ways to deal with these increasing stress levels with plants and with our food crops. So the first of our projects was utilizing Ceanothus velutinus, or snowbrush, which is a plant native to the Intermountain West. They grow up in the mountains, very, very desert, very dry, very, very low water soils. And yet they look very, very nice, and so there's a lot of call for them to be used as landscape crops. It'd be great, reduce the amount of water needed and be able to grow in very, very poor soils. However, one of the reasons that we selected this plant is that whenever this plant is taken out of its native environment and put into whether it's a greenhouse or whether it's someone's backyard, it does not do well at all. In fact, it almost always will die. And we theorize that might be due to the microbes that are present in their native environment, that when they're taken out of it, they don't have those microbes anymore. They're not able to kind of relate and aren't able to utilize and grow as well. And so to test that, what we did was we took these cuttings from Tony Grove, just down the canyon, got a number of cuttings from them, and then gathered the native soil as well, and then tried to propagate them. Took these cuttings. We have just normal propagation soil from the greenhouse, and then we did 50% mix of native soil and the propagation mix. And what we found was indeed that the cuttings that had these native soil in it had drastically increased rooting. So that first and foremost was great. We were able to actually propagate these plants. However, we wanted to understand what microbes were causing this effect. And further on, we'll kind of get into that, is what positive growth effects are they even having on these plants. And so that's what we did, was we took the soil around the roots themselves, and then the nodules from these plants. So these plants, these cuttings, as you can see, Ceanothus, is a nodule-forming plant. And if you're aware of common farming practices, soybean, peanut, all of these legumes have nitrogen-fixating microbes, very potent ones, that are in these nodules. And so that also kind of was part of the reason that we wanted to study this plant as well. But that's what we did. So we took those microbes that were in those environments, and then isolated them. That kind of moves on to our second area. Isolated them, got all of these individual microbes, got a whole bunch of them, and then tested them for a number of different growth characteristics. We had nitrogen fixation, we had indole acetic acid production, we had phosphate solubilization, we had iron chelation, or siderophore production, putting iron in a bioavailable form, ACC deaminase helping deal with heat and water stress, and then protease activity as well. And so this is kind of just an example. We did a number of different tests, but this is a very visible example, and I'll kind of pass these around. Oh, here, yeah, there we go. But I'll kind of show you all here. This is skim milk agar, which will show protease activity if these microbes are able to break down proteins, essentially, which would allow for could allow for a number of different effects for the plant itself. But you can kind of see that there is a halo around these ones that are pretty strongly positive. This is our control. Also, one of the versus the negatives, which didn't have this quality. So we tested them for a whole bunch of different plant growth promotion characteristics, and then shortlisted these 17 different microbes, basically taking the ones that showed the best across the board. And then we wanted, because again, the ultimate goal of this was to provide drought resistance and nutrient availability for plants in the increasingly more difficult environments of the world. So what we did was we took those microbes and tested them on Arabidopsis thaliana first, the nice model plant, and we did see increase in shoot biomass from all of them as compared to control. And then we tested the kind of the best ones on tall fescue, a grass, to see the effect. And we had a drastic increase of the fresh weight as well as tiller number from those that had these microbes applied. Last year, in fact, we went and did a test plot, a full wheat plot, just over there, using these microbes, and we're still finishing the data collection and analysis, so we don't have the data to present right now today. But so far, we've seen a pretty solid increase of fresh weight for the yield, and then also of, interestingly, depending on the microbe, nutrient levels as well was affected, and some were increased as compared to control as well. So there are a number of different potential benefits that we've found so far, but there's still much research that needs to be done, and we're still kind of working through that. We are also working to sequence these different microbes, specifically the best. You can kind of see these 4 that are really good, and then these 8 that are also beneficial. We sequenced generally all of them, but we want to do full genome sequencing for these really effective ones to see the actual characteristics. So what are the functions, what are the sequences that might be providing these benefits, so we can better be able to utilize these microbes as a way to supplement or replace natural chemical fertilizers within cropping systems. Any questions so far?
Where are you sourcing the microbes from?
Yes, so from the native environment. So these plants are from the native soil as well as from the ones that grow next to these cuttings.
And you're putting it on the tall fescue.
Precisely, yes. So once we have a microbe that we've seen has these positive growth characteristics, then we can grow it en masse. We did attempt to do a slew of them, but they were competing with one another. So that's something we need to analyze and better see if we can find some, what's the term? Compatible, exactly. Compatible microbes in the future, but for the ones that we've done so far, we just grew them en masse and then applied them whether as a seed coating, like seed soaking measures, or as a spray that you can just apply, or like a pour of the liquid microbes.